Greetings, Sir Marlon, and to everyone. We are the Group 9 and our topic for this video is all about preservation of fish genetics materials. My name is Gregor L. Gagara. Together with me is Patrick B. Diaz. Fish genetics preservation is all about safeguarding genetic materials from different fish species. It includes collecting, storing, and protecting the DNA, RNA, and tissues to maintain the diversity and sustainability of the fish populations. Importance of preservation of fish genetics materials. Number 1, species restoration and conservation. It is all about preserved genetic material aids in restoring and conserving fish species. It supports efforts to reintroduce endangered populations and initiate captive breeding, crucial for increasing population numbers and preserving species diversity. Number 2, breeding programs. Preserved genetic material is vital for implementing selective breeding programs in the field of aquaculture. It allows breeders to choose individuals with desired genetic traits such as disease resistance or the growth rate, improving fish stock quality and productivity. Number 3, disease management. Preserved genetic material helps understand and manage fish diseases. Studying genetic disease resistance enables strategies for breeding resistant strains or identifying susceptibility markers to specific pathogens. Number 4, biodiversity conservation. It is all about how maintaining genetic diversity within fish populations is crucial for their ability to adapt and remain resilient to environmental changes, habitat loss, and disease outbreaks. Preservation methods. Sperm banking. Sperm banking is about the practice of sperm banking in a fish where sperm from numerous males of a species are collected and preserved. It aids in conserving genetic diversity for cultured fish stocks, unique natural populations, and threatened species. Cryopreservation. Preserving fish genetic materials via liquid nitrogen involves freezing and storing genetic material like sperm, eggs, or embryos at ultra-low temperature. This process ensures long-term viability, allowing for the conservation of genetic diversity and the potential for future breeding programs, research, and conservation efforts.
Good day, sir. Today, I'm going to discuss my assigned topic, the tissue sampling and storage. Gathering small pieces of tissue from fish, whether they are live or deceased. These sample, which could be taken from fins or muscles, are then preserved for further analysis. There are preservation methods include freezing, drying, or immersing the tissue in ethanol or formalin to ensure their longevity. These preserved samples are stored at low temperature, typically ranging from -20 Celsius at -80 degrees Celsius. They can be stored in preservation solution for extended periods. Embryo cryopreservation is the method of freezing fish embryos during their early developmental stages to preserve them for an extended period. To do this, embryos are first treated with cryoprotectants, which are substances that protect cells from freezing damage. Then, specialized freezing techniques are used to cool the embryos to ultra-low temperatures. These frozen embryos are then stored in either liquid nitrogen tank or cryogenic freezers. 6 challenges in fish genetic preservation. The first one is genetic degradation, refers to the process by which genetic material such as DNA and RNA undergoes deterioration over time. This degradation occurs due to several factors including fluctuation in temperature, enzymatic activity, and exposure to light and oxygen. As a result, the genetic information encoded in this molecule can be lost or damaged, compromising the integrity of preserved samples. Essentially, genetic degradation can lead to a loss of a vital genetic information which can impact research, medical diagnostic, and other applications reliant on preserved genetic material. The second is resource constraints, refers to limitation in the ability of essential resources required for preservation efforts. These resources include funding, specialized equipment, and trained personnel. When resources are limited, it can be impede the establishment or upkeep of preservation facilities dedicated to conservation genetic resources. There may be gaps or inadequacies in genetic resources conservation effort due to the inability to allocate sufficiency resources to support these initiatives. The third is data management and accessibility in the context of genetic preservation, involve the organization, protection, and sharing of genetic data obtained from preserved samples. This process can be challenges to due to the complexity and volume of genetic data generated. To address this challenge, it is crucial to establish standardized protocols of managing data, ensuring its security and privacy, and promoting accessibility and sharing among researchers. By doing so, the utility of preserved genetic resources can be maximized, facilitating scientific research, collaborating, and innovation in various fields. Ethical and legal consideration in preserving fish genetic materials involve addressing concerns related to animal welfare, indigenous rights, and ownership of genetic resources. There may be ethical dilemmas surrounding the collection and use of genetic materials from fish, particularly regarding the treatment of animals and the potential impact on indigenous communities who relies on these resources. Moreover, navigating legal frameworks, governance access, sharing, and ownership of genetic materials can be complex as regulation vary across jurisdiction. These challenges can hinder collaborative research and data sharing efforts, highlighting the need of transparent and culturally sensitive practices when working with preserved genetic resources. The next is sample collection and storage logistics for fish genetics involve the establishment of appropriate infrastructure, facilities, and logistical arrangements. Challenges in this process include ensuring proper sample collection techniques, to obtain high-quality genetic materials, maintaining sample integrity during transportation to preservation facilities, and providing suitable storage conditions to prevent degradation. These logistics are crucial for preserving the genetic diversity of fish species effectively and ensuring that the stored samples remain viable for the future research and conservation efforts. The last topic is genetic erosion and loss of diversity, the gradual reduction of genetic variation within a population or species. This genetic diversity can occur due to various factors such as overexploitation, habitat loss, population, invasive species, and climate change. Genetic erosion has a serious consequence as it can diminish the fitness, adaptability, and resilience of population, making them more susceptible to environmental pressure and increasing the risk of extinction. Essential genetic erosion threatens the long-term survival of species by weakening their ability to adapt to changes condition and reducing their overall genetic resilience. That's all. Thank you.